Good morning, everyone. Thank you for joining us today for the Serious Talk webinar series. We've started this webinar series to mainly showcase the research of our customers and collaborators and basically discuss the challenges and the solutions to generating good quality data from degraded samples. So we've already had another talk, so please visit our website for our previous talks that we've given and other people have given on our behalf. Today we have Jordan Cheng, who will be presenting his work from his recent paper in iScience that used seriously. Jordan Cheng is a doctoral candidate at the UCLA School of Dentistry Oral Biology Ph.D. program. And under the mentorship of Dr. David Wong, his research is mainly focused on the field of liquid biopsy, which aims to solve unmet clinical needs in cancer treatment monitoring, early detection, and disease progression. He is the recipient of the National Cancer Institute's Predoctoral to Postdoctoral Fellow Transition Award, F99, which basically supports doctoral students with strong interest in pursuing careers as independent cancer researchers. And today he'll be speaking about his paper. His title for the talk is Exploring Ultrashort Single-Stranded DNA in Plasma. Welcome once again, Jordan.
So thank you, Varsha, for inviting me, and of course the whole Claret for inviting me to talk about my work today. I'm very excited, and everyone for attending. So I'm Jordan, and I'll be talking a bit about some of the work that we are able to do using the Claret Seriously Kit. But to start off, just about where we're in terms of our topic, cancer continues to be a very devastating condition, disease. And these may be numbers in 2020, but it continues to be a big player in 2022, up to the point that Biden's very interested in pushing forward the clinical treatments and also diagnostics. And he really wants to put forward a new effort into solving cancer issues going forward. So technology or even diagnostic protocols for cancer has changed quite a bit throughout the years. Traditionally in the past, after taking a tissue biopsy of a tumor, the pathologist will look at it under a microscope and use that to basically typecast it and say what type of cancer it is. But now, with the ability of sequencing and computation technology, we're able to get a lot more information that we can actually look at the genetic information of each cancer and not just how the type of cells look like. And this is particularly important because now, although you have a population of people with the same type of cancer, which is demonstrated in the top there, not all the cancers, even though they may be the same type, aka lung cancer or colorectal cancer, they're not all the same. If there are specific kind of molecular targets or genetic profiles, you can actually treat them differently. So here you can see that you have to categorize people based off the type of genetic information in their cancer, and then you give them a specific type of treatment. And if that type of specific treatment is not available, then you would give them something more general, which is what people have done in the past. So this is along the lines of personalized medicine. We're able to treat people in a very specific and personalized way. And here is a demonstration of if using lung cancer as an example. If they're given a certain type of mutation, there's actually different types of targeted treatment you would give them. So if they have an EGFR T790, you would give them osipertinib, for example. And normally, in order to get this information, people would do, of course, a tissue biopsy. And this is kind of the gold standard, but there's certain drawbacks of doing a tissue biopsy. First of all, it's surgically invasive. Anytime you put someone into surgery, there's a risk for complications. It's also only a single snapshot at a time. You only get what the tissue looks like when you capture it. And also, you may not get a full picture. You may maybe actually miss certain cells. So your actual sampling of the tissue may not be truly representative of all the different types of cancer that are available. So out came this concept of liquid biopsy, where perhaps you can use not just a tissue, but perhaps another type of a biofluid or biospecimen to get this type of information. And there are many types of biofluids we can actually sample, including saliva, sweat, blood, urine, CSF, and even your tears. And this idea is no longer kind of science fiction. It's been developed quite a bit. And there's actually many types of molecules you can look for example, blood. You can look at circulating tumor cells, which are cells that branch off or kind of jump off the host tumor and show up in the blood. You can even look at proteins or exosomes. But one particular one type of biomarker that's very interesting people are looking at is the circulating tumor DNA or cell-free DNA. So people started to realize that when the cancer cells or even all types of cells, they die, they actually release their DNA into the blood. And this DNA can be used to get that genetic information that we're so interested in seeing in the tissue biopsy. And you can use this DNA for early detection. You can use it to see if after treatment, if there's any residual cancer, and you can even use it to select the type of treatment. So you remember that chart I showed earlier, if you have one type of mutation, you'll give them this type of drug. So this is very well the possibilities with liquid biopsy. So in terms of the cell-free DNA, in general, it comes from cell death. So when the cells die, as they decompose and inside the blood, they'll end up releasing their DNA, and this will be chopped up by circulating nucleases. So when you actually sample the blood, you end up getting this very iconic pattern, which is shown in figure A, which is this peak at 167. So this is if you take the DNA in the blood and you were sequence it, you would get this peak. Now, where this peak comes from is actually from the DNA that's wrapped around the nucleosome. So if you look at picture B, for example, you see everyone recalls how DNA is organized in terms of chromosomes. But as you go further and further into the DNA, you actually have DNA that's wrapped around these nucleosomes in this pattern called beads on a string. So nucleases will digest everything, but there'll be DNA wrapped around the nucleosome. And this DNA is wrapped around the nucleosome essentially is 167 base pairs. So everyone's very curious, or most of the liquid biopsy is based off DNA that's wrapped around this nucleosome. And we see that 167 pattern. Now, over here, I just want to show how far we've gone since this concept has been put into people's minds. There's many companies that are interested in looking at the cell-free DNA to look for particular mutations. There are companies that have really invested in this idea. So it's no longer, as I said, like a science fiction idea. It's very well in the literature and soon to be in practice. I think many of these tests will be going through FDA testing soon, if not during the process already. Now, it's not that simple. It's not just because we have the DNA that it just works, because there's actually lots of challenges of liquid biopsy. Because you can imagine the blood has DNA from everywhere, as opposed to the tissue, which would have the tissue from the particular tumor you're interested in looking. So if you actually want to look for a specific mutation, you actually have to see the true mutation, which is demonstrated in red amongst all the different wild type demonstrated in black. And even white blood cells themselves, which contribute lots of DNA in the blood, sometimes they have their own mutations, which aren't real. So in order to actually find these mutations, you actually have to have lots of sensitive methods in order to see the true mutation. And as a result, you need to develop technologies to do this. So that's why this concept of needle in a haystack is a very common phrase in the liquid biopsy field, because you're basically looking for that needle, which is that mutation amongst all the wild type. Now, people have started to see different things. First thing they've noticed was that, interestingly, when you look at the cell-free DNA between healthy volunteers and cancer patients, a lot of the mutations are actually shorter. So in this picture here, if you look at a particular mutation, for example, let's say KRAS, you'll see that all the KRAS containing cell-free DNA actually has a shorter profile compared to in a wild type, because the wild type has your typical 167. And then furthermore, people have started seeing the same thing, that in cancers, those genes that contain mutations tend to be shorter. So there seems to be some pattern where mutated cell-free DNA is shorter. So it seems like looking at shorter strands or shorter DNA footprints is actually very useful. So I want to show this picture here, which is a picture of a chair with different shadows coming out of it. And the point of showing this picture is that everything we really see in the world, and of course in science, depends on the tools that you use. So if you change the tool, you maybe see different shadows. And ultimately, we want to see the truth, which is the actual chair. But we actually never really see the chair ever. We only see the shadows. So in a typical sequencing protocol, there's 5 major steps. There's sample collection, there's of course the blood, there's the DNA extraction, you have to prepare the library, then you sequence it, and then you do the bioinformatics. So using apples as an example, depending on how you change all these steps, they'll really affect what type of apples you end up putting into caramelizing your apple. So for example, in DNA extraction, in this picture, you have a device that sorts apples, and perhaps they want a certain size of apples. But if you have a very small apple, it's going to fall through here and not be selected. And similarly, in a library preparation step, if you don't end up putting an apple on a stick, they'll never be able to be used for the next few steps. So along this pattern, these 2 papers came out in 2016, where they said that if you use a single-stranded library, as opposed to a double-stranded DNA library in a next-generation sequencing pipeline, you'll get different results. And this is kind of what they showed. So over here, on the top is a double-stranded library preparation. And here, if you use a double-stranded library preparation, you can only incorporate certain types of cell-free DNA that's in the plasma. And you get this very traditional pattern that we see at 167. So what it ends up ignoring is it ignores single-stranded DNA, and it also ignores nicked DNA. But if you actually use a single-stranded library preparation, which separates all the DNA, you end up getting a lot more different types of DNA. You get the single strand, you get the nicked DNA, you even get jagged DNA, which isn't shown here, and of course you get the double-stranded as well. And they showed that you actually get a lot more DNA in this shorter area, and you also get more bigger spikes. So we had this hypothesis that perhaps there may be a different population of DNA that hasn't been observed before, and that's because of the techniques that were used. So we thought that perhaps there may be more DNA actually in the shorter region, because we're interested in the short because of the cancer thing, and also based off the library. So if we're able to combine both, we may be able to see that there's a different population of DNA besides the 160 mononucleosomal that everyone has been seeing before. So the 2 steps we wanted to focus on, which we thought contributed to this, was the DNA extraction and also the library preparation. So for the library preparation, it's quite obvious that we have to go for a single-stranded library method. And these 2 papers, they actually use this method from 2013 based off ancient DNA. So if you actually look at this ancient DNA protocol, which is a single-stranded library preparation, they have lots of different steps. And this is essentially a kind of a homebrew method. So we didn't really want to go for something like this. And we actually hoped at this time that there was a kit available. So fortunately, Claire actually has a kit for single-stranded library preparation. And that's why we ended up working with this kit, because it was a very simplified and refined version. They did all the QC we needed. We didn't want to go and make our own library, single-stranded library system. So, and also Claire also had a UMI technology, which allows you to correct for duplicates. This is where it was perfect, that there was already a kit available for the single-stranded aspect of our problem or our question, so to speak. So between DNA extraction and library preparation, library preparation is already good. We don't have to do that. So what we had to work on was on DNA extraction, because if DNA extraction loses types of DNA, we need to be able to fix that and catch everything. So our challenge was we need to find a way that best recovered short and possibly single-stranded DNA, and that we would pair it with a single-stranded library preparation to see if there was a different type of DNA or different population available. So in terms of the circulating DNA world, one of the gold standard kits is this Qiagen circulating nucleic acid kit, which looks like this. If you look at many of the papers, they'll have this kit used. So we kind of wanted to use that as our reference, so to speak. And we thought that magnetic beads might be a way to capture the shorter low molecular DNA. So we ended up developing our own magnetic bead extraction protocol. So over here is essentially what we ended up doing. It was through a lot of trial and error that we got a protocol that was able to capture low molecular weight DNA. And it essentially has the same steps of every extraction. There's going to be a protein digestion. There's going to be an isolation step. You're going to remove the proteins. You're going to clean it up. And ultimately, you're going to elute it. So as we were working on this and we got this magnetic bead method for extraction, we also realized that in that same Qiagen circulating nucleic acid kit, that there's another protocol called a microRNA protocol. So microRNA, of course, is RNA, but it has very similar traits to what we're looking for. We're looking for something that's short and also possibly single-stranded. And one of the biggest differences in this microRNA protocol is they use the same reagents, except they add in this lysis buffer, and they also add in a lot more isopropanol. So all the volumes are increased a bit, but this addition isopropanol seems to be one of the driving factors for being able to retrieve lower molecular weight material. So we ended up having essentially 3 types of methods that we could test. The first one is our Qiagen circulating kit, which I'll call Qiagen Classic or Qiagen C from now on. We also have a Qiagen microRNA protocol, or we call it Qiagen M. And we have our magnetic bead spry protocol, which uses the spry beads. But over here, it shows you that if you use the classic kit, you're going to capture the double-stranded, but single-stranded or small DNA is going to just fall through and not be captured by the silica matrix in the column. While in the microRNA, because of either the additional isopropanol and also more volume of the other extraction buffers, is able to retain this type of DNA. And we were going to combine it with the single-stranded library preparation. And in this case, we used Claret's technology. So this was the first or representative experiment we did. So over here shows the 3 different extraction methods and also a non-template control, which is during the library preparation, we just put water. But we see this observation. The first thing you see is this band at 300. And this band represents the mononucleosomal cell-free DNA. So one question you may ask is, you kept saying 167 for the past 15 minutes. Why is it 300? Well, during the library preparation, we actually add adapters and also UMIs and other things, which extends the length of the final product. So you actually need to subtract around 150, 169 from your product. So if you were to subtract that, you would get around 167. Now over here at 200 is where it got really interesting. Using the Qiagen Classic or the Qiagen C protocol, you didn't really see much, but using the Qiagen M and the spry, we found this band at 200. And of course, if you subtract 150-ish, you get around 50 base pairs. So there's something short that was found here that's not found in the Qiagen C, and it wasn't part of the library system, because if you do a control, you don't see anything. And on the very bottom here is the adapter dimers, where essentially the adapters, the purple and the orange, they kind of bind together because there's nothing to be found. So that's always going to be there. So we were very excited to see this band at 200. And this is a reproducible phenomena. It wasn't just like a one-time thing. So we did it amongst several donors. In this case, it was 10 plasma. And we found that it was very repeatable. Using these 2 methods, you always got a band, while with the Qiagen C, you never got a band. So it seemed to be not just a one-time fluke. So we proceeded to sequence it, and this is what we ended up getting. So here, Qiagen C, you get that 167 peak profile that we're very familiar that we saw previously. This is around 167, and then you get all the shorter DNA. Now, but if you use the Qiagen M and the SPRI, you get this peak at around 50, maybe it's a little bit longer, maybe 52. But this was seen in both the Qiagen M and also the SPRI. And when we looked at how the DNA, the coverage of the DNA, using more of a kind of a broad approach, you can see here the solid color represents the monocromosomal, so all the DNA between 100 and 250, and the black represents what we call the ultra-short cell-free DNA, anything from 25 to 100. So the coverage amongst the human genome was actually quite even. There wasn't particular one, well, in this case, one chromosome where all the reads went to. And this was important for us to see because we were always concerned, could it be an artifact where all this ultra short is actually from one chromosome, but it seems to have reads that went to every chromosome. Although if you look over here, there may be certain areas that they are enriched. And if you look even deeper, there's actually certain peaks that are enriched. But in general, all chromosomes seem to have ultra short reads. In terms of another control experiment, we just wanted to see if everything was okay. So we extracted DNA or whatever's available from just TE, which is tris EDTA. So basically no biological aspects. So we put that into the extraction and we made a library, and we weren't able to represent that 50 nucleotide, 50 base pair peak, let alone the 167. So this was kind of a good check for us. And of course, if we're doing a microRNA kit for Qiagen, we have to there's a concern if this is RNA. So in this experiment, we did RNA digestion on the DNA after we made a library. So if you do a RNA digestion, this is the case, this is with the RNA and RNA digestion and without, we weren't able to remove that band at 200. So it appears this ultra short band isn't RNA. But also at the same time, the Claire kit shouldn't actually make libraries out of RNA. So in this, we kind of concluded that it's not actually RNA we're seeing, it's actually DNA. So these are kind of our two conclusions. We found that using this combination of tools, a low molecular weight extraction plus a single stranded library kit, which we call ultra short cell-free DNA sequencing, we're able to reveal this new population or a different population at 50 nucleotides that's quite distinct from the 167, and it is not RNA. So another thing we want to look into was some of the characteristics of this ultra short population. So there's already been kind of clues that perhaps this shorter DNA is actually single-stranded because the shorter stuff here wasn't revealed unless you were able to do a single-strand kit. But interestingly, from a clinical perspective, people were interested in looking at cell-free single-stranded DNA for some reason. There's actually two papers were published where they wanted to look at the ratio between single-strand and double-strand. And in these projects, they used a qubit to measure single-strand, which is actually not that accurate for single-strand. But the fact that they were looking at single-stranded is kind of a coincidence, because we also wanted to see, is this DNA single-strand or double-stranded? So that was kind of our next question. What kind of strandedness is this ultra-short DNA? So in order to do that, what we did was we kind of did an intervention between steps 2 and 3. So after we extracted the DNA using our low molecular methods, we intervened and saw what would happen after library preparation. And the reason we had to do this was because we don't actually have sensitive methods to visualize the DNA after we extract it. There's no gel that's sensitive at this time for short molecular DNA. So we actually have to make a library out of everything at the very end. So what we ended up doing was we tried different types of enzyme treatments. So in this case, we tried this enzyme, which is a double-strand DNAs, which what it does is it digests all double-stranded DNA. So theoretically, what should be left for library preparation is something that's single-stranded. Opposingly, we did S1, XO1, which are both enzymes that digest single-stranded DNA. So theoretically, what should be left should be double-stranded. So what we did here was we did the experiment. And the first thing we did is, of course, we used our controls. So we had controlled DNA, which is a lambda sequence, just to make sure our enzymes were working properly. So our first column is essentially our control. You put in both your DNA and you're not doing any digestion, so it retains itself. And this is also, and over here is the cell-free DNA from the plasma. So you get your monochromosomal cell-free DNA, the 167, and you also see your ultrashort. But after you digest it with the double-strand specific enzyme, in the control, you remove the double strand, which is correct, and your single strand remains. Now, for the actual biological DNA, it appears after you treat it with double strand, you remove a lot of the monochromosomal, which is known to be double strand, but the ultrashort remains. This gives some, I guess, a clue that maybe it's actually single strand in origin because it's able to resist the enzyme. Opposingly, if you do the single strand specific enzyme, which will delete the single strand DNA, the monochromosomal is able to survive, but your ultrashort disappears. So kind of as a conclusion of these results, it appears that ultrashort has some single strand properties, and this was reproducible over 3 donors. And after we sequence it, you get the kind of the representative shapes. But one interesting note that you can see here, the dotted line is your control, so this is what we expect. And after you do the double strand digest, only the single strand remains. And after you do the 2 enzymes for the single strand, the ultrashort disappears. But I just wanted to say that the S1 enzyme is very interesting because the S1 enzyme appears to remove the jaggedness of the pattern and also shifts everything a bit to the left. And the reason for that is because S1 doesn't just remove single strand DNA, it also removes kind of the ends of overhangs. So that's why it may have a very unique pattern, but it probably tells you a lot. I think we should actually just explore S1 a little bit more. So another way we could attack this problem is to actually utilize one of the initial steps in the Claire kit. So how it's able to see single strand is there's a denaturing step in the kit. So what it does is it heats up all the DNA and they all separate into single strand. So here you can see the different types of DNA. The single strand, there's nick DNA, which is a nick in the middle, and double strand. And after you heat denature, they all become essentially single strand, and they're all able to be incorporated in the library, so into the final library. So what would happen if you actually didn't do this step? Theoretically, only some types of DNA should be included. So this is kind of what we did. So here's the traditional, or the control is the heat shock. If you do the heat shock before the single strand library, you should include all the DNA. You'll include the single strand and the double strand. But if you bypass the heat shock and skip it, theoretically only the single strand should be incorporated because the double strand wouldn't denature and separate. So it should exclude double strand. Similarly, if you do the really traditional library preparation, which is a double-stranded DNA library preparation, where there's no heat denaturing at all, it should only incorporate double-strand DNA. So all the single-strand DNA should be excluded. And one of the questions people ask would be like, is this DNA from nicks, from nick DNA? So if you repair it using a repair enzyme, if the single strand was from those nick DNA, then they should theoretically disappear. So we tested this similarly, like the last experiment, and this is what happened. So over here is the first column, so to speak, is once again the control. You see everything is kind of intact. But if you skip the heat shock step, then ultimately in the library, in the control, only the single strand gets incorporated, which is kind of what happened here. When we did it with the plasma DNA, we saw there's still some monochromosomal that remained, but it dwindled a lot more. But your ultrashort kind of kept its signal. So this is some proof that there's some single strand properties to this ultrashort. And if you do the double-stranded library preparation, which only incorporates double strand, you see only the double strand remains, but the ultrashort or the single strand disappears. And with the actual plasma DNA, only the monochromosomal remains, but your ultrashort DNA disappears. So this is, once again, more proof for single strand. And when you do the nick repair, nothing really changes. This doesn't disappear. So once again, it was repeatable amongst multiple donors, in this case 3. And when we did the sequencing, we got the supportive results that if the dotted line is your control, after you do the heat shock, the most, which is orange, the mononucleosomal goes down, but your ultra-short signal goes up. And when you do the double-strand library preparation, you don't get any ultra-short incorporation. So all of the DNA at 50 no longer remains. And with the nick repair, it seems it affects the jaggedness of the pattern, but it doesn't remove the ultra-short DNA. So it doesn't appear that ultra-short DNA is from actually nicked DNA. So this is just showing all the experiments we did and how we ultimately got those graphs. And that not only was it shown using the Qiagen M extraction, I only showed you the Qiagen M, or the microRNA, the same results occurred with the spry extraction as well. So regardless of extraction, the same digestion experiments were reproducible, showing, I guess, both those methods do capture the ultra-short, and they're both, the ones they capture are single-stranded in nature. So it kind of includes conclusion, ultra-short DNA appears to be primarily single-stranded and not from nicked DNA. So since we've done that work, we got it published in iScience, but we actually weren't the only ones to see this pattern. So over here in the middle is this very nice kind of summary diagram that was published in a JCI paper by Thierry, which is actually a scientist who studies cell-free DNA quite a bit in Europe. But it kind of summarizes all the different peaks and where they're from. But it just shows the iconicness of the 167. And what we're seeing is now this peak at 50 that can kind of update this figure, so to speak. So we saw it and we published it, but as I was saying, we weren't the only ones. A group in Japan actually saw the same thing, and published a little bit earlier, actually. They also found the peak at 50, and they said it was single-stranded. And their methodology was using phenochloroform. And a very prominent group in Cambridge and Amsterdam, they also saw the peak at 50. They called it, or they may not say single-stranded in title, but they said it's ultra-short. And later in their paper, they say it's also single-stranded, and they used a magnetic bead protocol. That's why the MB. And also another group that we kind of work with, they also found it at 50 nucleotides. And their method, they used a hybrid probe, which is a probe that they kind of threw into the plasma and fished at all the sequences. And because they could only bind to single strand, it should only capture single strand DNA. And as you see, their 167 is actually really low. And the reason for that is because their method isn't good at capturing mononucleosomal cell-free DNA. So this data kind of makes sense. So we're kind of happy to see that not only did our groups, was able to observe something ultra-short, but multiple groups were able to see it. So it's not just a kind of an artifact of our whole system. And all these methods are slightly different. So it seems to be universal in, of course, different types of plasma. So this is kind of where we are right now. We observe that there's ultra-short cell-free DNA. And logically, people will be asking two types of questions, which is, what is this biological significance? Like, where does it come from? Is it like a special biological mechanism we haven't explored? And of course, the whole intro was on cancer. And it's like, what can we do with it? So right now, it's kind of where we are too. We've figured out that there is ultra-short, and now we're going to explore these two arms. But in terms of where it's from, one of the hypotheses people would say, like, well, your ultra-short probably comes from your mononucleosomal cell-free DNA. Because even though we see 167, maybe as those nucleosomes get destroyed, it gets further cut down. But another possibility is that it's not from mononucleosomal breaking down. It could be actually a totally different mechanism altogether. And some of the clues that suggest that is if you actually look at the sequences contained between these ultra-short and mononucleosomal, between all three extraction methods, they're actually extremely different. There's actually not that much similarity. And one way to actually further look into this is to use this peak collar. So this is the bioinformatic protocol. But this is the thing, it's like we have no idea what we're actually looking at. So all we can really do is scan our bioinformatic files to see if there are certain sequences that appear enriched. Even though previously I showed that it's spread evenly amongst the chromosomes from a very broad perspective, if you actually narrow it down, there's actually certain regions that are enriched. And all the sequencing we've been doing is non-targeted. It's completely, it's like equivalent to whole genome sequencing. So anything comes is what we see. So we basically use this protocol that we borrowed from the ChIP-seq people. The ChIP-seq people are used to study the DNA wrapped around proteins that they are able to capture, but it's able to see peaks. So we use the same aspect for our work in order to see if there's any in terms of sequences that are very prominent in our files, and using Homer, if it could tell us what it thinks it is. So of course, we're kind of held hostage to whatever Homer says, but this is where we have to start off for now. So this is actually one set of interesting data. So over here, Homer is able to categorize sequences based off different categories. These categories are, is it some sort of like rRNA, RNA? Is it a non-coding RNA? Is it a untranslated region or promoter or an exon? And it's able to tell you if there's a peak, what type of a sequence is it? So here we compared, we split the DNA based off their size, and we had the mononucleosomal cell-free DNA based off the 3 different extraction methods, Qiagen C, M, and SPRI. And we also split bin the reads based off their shorter size, which is Ultrashort, for the 3 methods as well. So we actually zoomed in on this area a bit. Actually, based off here, the first thing we observed is that the mononucleosomal and the Ultrashort have like a very different pattern. There appears to be a lot of intergenic sequences in the mononucleosomal, but not so much in the Ultrashort size reads. And the Ultrashort reads actually represent the genome reference a lot more. But if we zoom in here, you see it's a lot more obvious too. What we actually observed is that there's actually a lot more promoter regions in the Ultrashort and a lot more exonic regions, or maybe equivalent to the regular genome found in the Ultrashort. So it tells you that the DNA sequences and the Ultrashort are actually quite different for the mononucleosomal. So either that's just how the breakdown pattern occurs, or maybe there's a different mechanism that manifests or this type of DNA in the plasma. And if you actually look at the specific genes, so here are some of the sequences that are called by Homer, such as STIX and MMD2. And here we have 4 different healthy donors, and there are some similarities amongst them. But what I wanted to stress, this is a lot different from the ones you see in the mononucleosomal cell-free DNA. So they appear to be actually two very different populations. So this is kind of where we are right now. We're working on these 2 directions very fervently. We're really curious in the biological significance of the Ultrashort, and we're also doing looking if the Ultrashort is useful for clinical applications. Can it be used to fix or to aid in that needle in the haystack problem that liquid biopsy is facing? Because that's what got us interested in this to begin with. And now we're going to go back, especially if they're different sequences, they may have different properties that could help us. But I actually don't have any data to show right now, but we're actually working on this right now. And probably in the future, we'll share that as well. So just want to thank, of course, Claire Bioscience for inviting me to speak today. And of course, everyone in the lab at UCLA and our collaborators in Taiwan, and also at NIST that kind of made this work possible. And of course, the funders as well. And of course, everyone for attending today, for listening. So I really appreciate it.
Thank you, Jordan, for talking about your work. It's really fascinating and something that we think about daily, and I'm sure several folks in the audience also think about this quite often. I see there is a question already. It's from Jocelyn. She asks, what was the input into the library prep? Was this mass based on double-stranded DNA concentration? How did you quantify your DNA extracts?
Yeah, so that's a really good question, because it kind of, what we actually ended up doing was we did everything based on volume. So if we took 1 mil and we extracted it using our extraction method and we eluted it into 40, and then we put 18 into the library. So it's completely based off volume. And we didn't want to use the qubit to measure double-strand DNA. We actually have some experience where that's actually not a very good way to do it, because it ignores the single strand. So what we're actually looking to do now is find a way to quantify the ultrashort, which is actually quite hard because none of the qubit methods, of course, nano drop, they're able to actually measure the ultrashort. So our initial work, we just did it based on volume, but we're very interested in being able to quantify the extracted DNA, because then we can even answer questions like, what's the normal type of cell-free DNA concentration or ultrashort concentration in healthy versus cancer versus if you're running versus if you're stressed. But at this time, we don't have the technology to do that. So we just did it based on volume.
Yeah, if you do figure out how to quantify double strand versus single strand, we'd be happy to know too, as several folks on this webinar. The other question is from Meredith Carpenter. She asks, do you also try to map the ultrashort fragments to other genomes, example, microbial sequences?
So we didn't map the microbial sequences, but we checked the concentration to mitochondria DNA. And we find that mitochondria DNA is a very small population, like 1%. But we've done like different cell type. We've done the same kind of work with like saliva mapping to microbial sequences. Is also, I think the saliva, it plays like a big role, but in the blood, we didn't map it to microbial sequences. I think we could try doing that, but we shouldn't expect too many bugs in our blood.
Yeah, I actually had a couple of questions about, mainly about pre-analytical variables. So we know that plasma DNA, once you extract it, can be very unstable, prone to degradation. Did you do anything special to make sure that your DNA was stable? Because there are blood collection tubes out there which, maintain integrity and so on.
Yeah, so I have the data. I don't know how to show it, but then we tested, because most of the data I showed was blood collected in K2-EDTA tubes, but we also did the same experiment with Streck tubes, which is right now considered the gold standard for preventing degradation and for cells to, explode or lyse. So we did the same experiments with the Streck tubes, and we were still able to see the same pattern. So that's kind of suggests that the ultrashort isn't coming from rapid degradation of monochromosomal cell-free DNA. But we didn't do any other further treatment. After we extracted it, we just froze it.
Got it. I think there are a couple of questions from Chris Troll, whom you know well. One is, do you think it would be beneficial to remove the mononucleosomal DNA from your libraries to increase reads from the ultrashort DNA? And have you tried that? Like, what is the distribution? Yeah, go ahead, answer.
Yeah, so I think that would be beneficial because what you see in the figures is the ultrashort and the monochromosomal, but you also see, you actually may not see reads going to the genomic DNA that we weren't able to remove. So it would be nice to invest all the DNA, so to speak, into the ultrashort. And we actually have a method to do that. We're able to actually physically separate ultrashort and monochromosomal. So when you have that separation and you make a library, you have a very ultrashort enriched library. If you want to only look at that types of DNA. So in the future, if it's very useful, just look at that, then that's actually a method that you can invest all your reads into ultrashort.
So sort of a follow-up on that. So in your preliminary information that you shared, where you see that there are unique genes that are enriched, would the idea then be that if you see an enrichment of, cancer-specific signal in the ultrashort, you would end up using a method like this for that purpose specifically?
Yeah, exactly. And then you can probably even, if you can do that for the extraction method, you may not even need to sequence. If you have methodologies
Right.
like modified PCRs that can look at the kind of the extracted DNA, then you can really narrow it down to what you want to look at. But right, in the future.
Yeah. I think Chris has another comment saying dual-sided spray post-index PCR would do that. So the idea is you make your library and then you do the selection for the part that you need.
Yeah, I think that's a good methodology.
Yeah.
Of course, it needs a lot of tinkering, and I guess you have to check every time to make sure what the best settings are in order to get that.
Yeah.
But the way we're able to establish to actually do it before that, but then we don't have to actually worry about any types of size selection.
Yeah, I think the advantage with post-index is you have a lot more DNA, so you won't have to worry about losses at that point. Yeah, but as you said, tinkering will get you where you want to go. All right. If there are no other questions, please join me in thanking Jordan once again for this wonderful talk and for all his inputs about this unique class of DNA within plasma, which is gaining a lot of interest, as he just mentioned. We're really happy to see that, seriously is helping him with this kind of work. Thank you everyone for joining. We will have this video available for viewing later. Thank you so much.